All right. So, hello everyone, welcome to the webinar. I hope my screen is visible and my audio is also working fine.
Yes, Shrigori.
Thank you. Okay, I can see that participants are still joining. We will start with the webinar in about 10, 12 minutes. We have started the session a bit early so that participants can join and we can start maybe briefly introducing ourselves so that we can learn about your educational background and research interest. So, shall we start with Chaitra? Hi, Chaitra. How are you?
Hi, ma'am.
Yes. How are you?
Fine, ma'am. What about you, ma'am?
I'm doing good as well. So, could you please tell us where you're from and what is your educational background?
Actually, I'm from Bangalore and I'm pursuing the undergraduate degree in biotechnology.
Okay, perfect. And have you been part of any of our previous workshops or training programs?
No, ma'am, this is my first time.
Okay, okay. All right. Thank you so much, Chaitra.
Thank you, ma'am.
Okay. Now, can we hear from Iba? I hope I'm pronouncing it right. Please feel free to correct me. Iba, can you hear us? Okay. All right, then let's move on to the next participant in that case. So, Vasanta Kumar. Hello, Vasanta Kumar. Could you please briefly introduce yourself, where you're from, and what is your background? Okay, he's texting in the chat box. So, Vasanta is from Tamil Nadu. Perfect. And what is your educational background? Are you an undergrad or a master's student or a working professional? Oh, yes, I can see Iba has raised hands. Yes, Iba, go ahead. Can you try to unmute? It seems there seems to be some technical issue. I can see you have raised your hand. Yes, Iba.
Good morning.
Yes. Good morning,
My
Iba. Now
name is Hajar Ziadi. From Algeria. I'm PhD student in molecular and cell biology. My research is talking about antibiotic-resistant Enterobacteriaceae associated with infection diseases.
Okay, perfect. Thank you so much, Iba. So, have you previously been part of any workshop or program, or is it the first time?
No, this is my first one.
Okay, all right. Thank you so much, Iba.
Thank you.
All right, I can see Vasanta Kumar has also texted in the chat that he has completed his MTech in computational biology from Pondicherry University and is currently working in as a research fellow at IIT Guwahati. That is perfect. So we have two participants from the research background. So that is good to know. So mainly we during this webinar, we will be guiding you through the different learning resources available on the OmicsLogic Learn platform and guide you through the different topics that based on which there are coursework, how you can access them, the as well as if you have any questions based on which training resource might be the most suitable for you, then we can also take that up during the session. Okay. So I guess participants are still joining. We will wait for a few more minutes and then start with the session. All right. Okay, so I can see several more participants have started joining. So for those of you who have just joined, welcome to the webinar. We will be starting within about 2 minutes, 2 to 3 minutes. We are waiting for rest of the participants to join. And in the meantime, it would be great if you guys can introduce yourself. You can let us know where you're from, what is your background. You can either type it in the chat box, or you can also unmute yourself and introduce. This will help us to understand how to tailor the webinar for today. So if you have a lot of participants who are complete beginners in bioinformatics and data science, we can keep the session a little bit more simpler. But if we have a lot more research-oriented students, in that case, we can maybe align the topics accordingly. Please do also let us know where you're from and what is your background, and we will be happy to read it out and get to know. Okay, so Sadat is from genetic engineering and biotechnology background. and is from Dhaka University. Perfect. Thank you so much, Siddharth. And he's an undergraduate. Okay. So we have now, I guess, 2 undergrads. Have you been part of any of our previous workshops or programs, or are you new to OmicsLogic? Can you tell us that also? Okay, Ryan is from IMS Engineering College. Oh, yes, we did have a couple of students from IMS last year for an hour workshop. Are you also part of it, Ryan? Or are you joining with us for the first time? Okay, so I guess we do have a couple of participants who have joined just now. So then I guess we can start with the webinar. And meanwhile, you guys can definitely introduce yourself in the chat box. It will be definitely great to know you. Now, just before we begin, the session will be recorded and we will share with you the recording in case you miss out on any points or you would like to review it. All right. So without delay, I'll start with the webinar in that case. So Jivisha, if you could start. Okay, perfect. So hello everyone, a very warm greeting to one and all. I'm Shrigari and I would like to welcome you all for the webinar on specialized tracks and hands-on projects with OmicsLogic. So in this webinar, we will introduce you to the various structured learning resources that are available on the OmicsLogic platform. We'll also give you a detailed overview of how these coursework and resources are designed to help students, researchers, and professionals build expertise in omics data analysis. And you will also learn about the flexibility and benefits of our training resources, how it gives you access to the courses, hands-on exercises, and real-world data set. And finally, towards the end, if you have any specific questions about the coursework or learning outcomes, or how even this training will align with your research or career goals, we can definitely take it up towards the end. Okay. Now, before we begin, I would also like to take a brief moment to introduce you to the team behind the program. So we are India, an US-based bioinformatics and data science company, and we have had the wonderful opportunity to collaborate and partner with esteemed universities and industries across the globe. Now, as a part of our mission, we are constantly working with regional and international research mentors that are helping refine program logistics and adapting them to the needs of students and researchers around the world. Now, to meet the ever evolving demand of skilled bioinformaticians and data scientists, we have created innovative training and research offerings, including online courses, training programs and workshops, research internship and fellowship, research services for faculty, scientists, as well as a dedicated online degree program. Now, we also have a few upcoming online events that are workshops. They are live mentor guided sessions with hands-on practicals involved. So if you're someone who's interested to explore workshops, these are a few details. So the very first workshop that we have upcoming is starting right from next week on Monday, which is on cancer genomics, and it will be a 4-day workshop. And from March 17th till March 20th, we have another 4-day hands-on workshop on metabolomic data analysis. And finally, we also have one more workshop on March 24th till March 27th. If you're a plant biotechnology enthusiast, then we have a workshop on plant biotechnology for RNA-seq data analysis using R programming. All right, so if you would like to explore any of these workshops, you can definitely put a comment in the chat box, or you can get to us towards the Q&A part, and we will be happy to share more details. Okay, now the main objective for today's webinar is to discuss about the OmicsLogic coursework and tracks that are available. Now, what do these exactly contain? So here, if you can see on my slide, you can see an overview of the coursework, structure of a coursework. So this includes text and visual aids such as diagrams, infographics, short tutorials to simplify complex concepts in bioinformatics and data science. There are also practical demonstrations that are added to provide and give you a hands-on learning experience. There are also coding exercises to help you develop your technical skills. Now, we have also incorporated open source tools and technologies in the coursework, which I'll be just covering in the next few slides. And at the end of the coursework, as well as in the programs, students can earn digital certificate, which you can later add to your resume on your LinkedIn profile to enhance your professional credentials. So we will explore all of these coursework much more in depth in the later part of today's webinar. This is just to give you an overview of what you can expect in the upcoming part. All right. And all these courseworks are also fully automated and compiled into detailed lessons that then, and many times students, they require some mentor support as well as some video lectures to supplement their learning. So to do that, we also have a curated collection of training programs and workshops. And workshop was one of this that I covered in my previous slide. So these training programs and workshops consist a combination of lecture videos and mentor guided sessions. The training materials are always designed to align with the modern industry relevant curriculum, and it constantly gets updated so as to ensure that all the students are able to gain practical skills that are applicable in the current academic and professional settings. Now, we also have an online progress tracking system to help monitor the students' progress throughout and assist them wherever required. And this has two aspects. That is, as I mentioned, one is the training program and one is the workshop. So the training program comprises of, the workshops consist of live mentor guided sessions, while training programs are mostly asynchronous in nature, which are mostly suited for working professionals who are occupied and they might want to study independently by going through the resources, right? Now, here is a brief summary that I was telling about that covers various open source tools and technologies that are covered throughout our programs and coursework. And these are common scientific tools that are used by researchers worldwide. And there are many more tools that we cannot cover through slides, but we will be taking them through during the later part of the session. And this is evident from the thousands of students who have gone through the program successfully and are now working in reputed firms. And as you can see, these are some of the feedback that we have received from previous batch of students, right? So now I'll just quickly take you to the OmicsLogic Learn platform, wherein all these resources are put together, and we can give you a brief demonstration of how you can navigate the platform and explore all of these resources. So now let me quickly reshare my screen. So some of you might already be familiar with the platform, but for those of you who are new here, so OmicsLogic is an online learning platform wherein we have curated all the coursework, example projects, as well as we have a dedicated research program where students can apply all of their learning into real world projects, right? So here is the link for the platform. I'm just sharing it so that you can keep it handy. And if you're on your system, you can also navigate it along with me, right? So for those of you who are visiting this page for the first time, if you want to explore the pages, you'll be required to create an account. It is a free process. You need not worry about it. Yes. So this is the sign up page when you click on the login. For those of you who have to create an account for the first time, just click on this. And you can enter your name, email ID, password, and you can create an account. Now, for those of you who already have an existing account on the portal, just click on login now, enter your existing email ID and password, and you can directly sign in. Okay, so with that, I'll just pause for a couple of seconds for participants to create an account or log into their existing account. But once done, just let me know a yes in the chat box so that I can proceed with the next step. If you're facing any technical issues while signing up, you can also definitely put them in the chat box and we can help you and we can help look into that. Yes, for those participants who already have an account but don't recollect their password, there is also an option to click on Forgot Password, and you'll receive an email to reset your account, and then you can directly log in. And in my next part, once you guys have logged into your account, I'll be just showing you how to set up your profile and navigate the resources. So just let me know once we are good to go. Just put a yes in the chat box, and I'll move on to the next part. Okay, so a few of them have responded yes. Okay, thank you, Somit, Ragia, Yusuf, Enoch, Kausar. Thank you so much, everyone, for responding. Thank you, Chaitra. I guess most of you have started logging in. Then I'll proceed with the next step. I'm going to log in with my existing account. So once you log in, the very first interface that you see is the course library section, where we have curated all the coursework and example projects on different omics fields like genomics, transcriptomics, metagenomics. We will explore all of this in just a while. But before we do that, the very first thing that you have to do is to set up your profile. So here you can see a blue bar that displays your name. Just click on that and it will redirect you to your profile settings. So here you can click on the profile button, you can upload an image, make sure that your name is in proper capitalization and there is no spelling error. You can also add a brief bio about yourself, like your educational background, your research interest, and a link to your social media handles. Okay, so once all of this is done, your profile will be 100% complete. Now, as I mentioned in my slide, you also have a online tracking system to check in on your progress. So this is where we can see under the activity tab. And there are several other tabs on your profile based on how you're progressing through the coursework. All of that will be displayed here. Okay. So now moving back to the course library section. Here, I would like to pass on the stage to one of the mentors of the team who will be guiding you through the different resources available on the course library section and guide you through the process. So over to you, Javisha.
Thank you, Gauri. Hello, everyone. Welcome to today's webinar. And I am Divisha, and today I'll be talking about our subscription model and the course track. So I hope my screen is visible and I'm perfectly audible. Just let me know.
Yes, it is working fine.
All right, thank you. So to access the courses, we go to course library. And once you are here, you can see we have listed out various course categories based on your area of learning. For example, you can see bioinformatics, genomics, transcriptomics, machine learning, Python for omics data analysis, R for omics data analysis, and so on. All right. And if you scroll down, you can find the courses that are available on the portal, and you can click on these numbers and browse through it all. And on the right side of the screen, you can also see another course category, which you can click on and browse through the courses. So I would like to begin with bioinformatics course category. And when you click on it, you will find the first course, which is course 1, Introduction to Bioinformatics. Now, this particular course is a free course, and you can choose to go through this after the webinar is over. And it contains 8 lessons in total. Okay, and if you are new to bioinformatics or know very little about it, then you can go through this. We start with the introduction to bioinformatics, then what is the data, how is data analysis done in bioinformatics, uh, what is the big data in bioinformatics, and its applications in healthcare and translational bioinformatics, and we end with genomics, transcriptomics, and metagenomics fields. All right. Now, if I open up a lesson for you, then you'll see that we have the concepts discussed in theoretical or textual form. And in the lesson, you will find images, diagrams related to the concept that we are discussing. And to get a better understanding, we have added these for you. And we have also added activities and assignments at places. And in some sections, as you can see, for example, here's a structure of faster file, and we have described it for you. So this is all for you to get a better understanding of the topic that we are discussing. And all of the lessons that are present in the courses follow the same pattern. All right. And again, you can see the images. So this is all for your better understanding. Now, at some places, you'll also find short conceptual videos to explain you a particular concept. And most of the lessons are ending with the quiz. So once you are done with the lesson, you can test your knowledge here with the quiz. All right. So this is a course that you can go through after the webinar. Now, the next category I would like to discuss is genomics. Once you click on it, and it will show you all the example projects and courses that are related to genomics. And if I open up this course for you, you will find around 17 lessons. Okay. And these lessons are created in a very structured format. So we start with very beginner level topics. For example, here DNA structure and variants. Then we introduce genomics, and then we move forward with next generation sequencing techniques that are used in genomics. And then we start with the genomic data analysis. We start with variant calling, sequence alignment and phylogenetic analysis, copy number variation analysis, and how to do genomic analysis with the Galaxy tool, ANOVAR for variant annotation, and how to use CBio portal, and how to employ R programming for genomic data analysis. All right. And for example, if I open up this particular lesson for you, you will find that we are discussing tool known as integrative genome viewer, in which you analyze your VCF files uh, after doing a pipeline for variant calling. And then we discuss databases for variant annotation like Ensemble, DBSNB, and Glenva and Cosmic. All right. So as you can see, the courses are in a very structured format. This is just one example. But if you check our other courses, then you'll see a similar format in all of them. And again, if we click on transcriptomics course category, then you'll find example projects and courses related to transcriptomics. And this is one of another courses that is very popular with our learners. And again, it has 17 lessons. And we start with very basic topics like central dogma to RNA, what are the RNA quantification techniques, and what are the NGS techniques used in RNA-seq. Then we start with the workflow of transcriptomic data analysis. Now, the special thing about transcriptomics course is that it follows a workflow of RNA-seq data analysis. First, we are pre-processing the data using Linux and Galaxy. Okay, then we are doing the quality control using FastQC. We are normalizing our data. Then we are conducting statistical test on that data that the results that we get from normalization. And then we proceed with the differential gene expression analysis and enrichment analysis, functional characterization of the genes, and at last we cover regression and factor analysis and machine learning methods on our RNA-seq data. Now, in the courses, we have added open sources tools like Linux, Galaxy, FastQC, and we are again using R coding for the analysis. All right. Now, in the transcriptomics course, you can see that some of the lessons are repeated, but they are not actually repeated. They are just telling you how you can do a particular step by different methods. For example, in BGE analysis, here we are using different open sources tools. And in this, we are particularly using R programming. All right. And this is how the transcriptomic course looks like. Next, I would like to show you Python category and R category. Okay, when you click on Python, you will find two courses. One is Python course 1, which is a beginner friendly course. And if you are from core biology background and don't know about coding, which is again integral part of bioinformatics, then this particular course will be a good start for you. And similarly, a similar course will be there for R programming as well. So here we start with Python installation, basics of Python, where we will discuss variables, data types, and various operations, functions, modules that are used in Python. Then we discuss Python packages and Python packages that are specific to omics data analysis. All right. And there is a little two lessons which are hands-on, in which in the first you are learning about how to read DNA using Python and how to find DNA replication and reverse complements. Right. Now, um, just a second. Okay. Now another course is advanced course on Python, which is covering machine learning using Python programming. All right. And we start with data wrangling and visualization, descriptive statistics. We are covering dimensionality reduction methods like PCA, MDS, NMDS, clustering, and which includes k-means and hierarchical clustering, supervised analysis, which will include classification. And then we have data visualization using t-SNE and UMAP, uh, dimensionality reduction using deep learning, and how to plot with Plotly and predictive modeling with deep learning, and t-test and heat map uh, using Python. All right. And I'll show you this lesson and tell you how it is different from the lessons that we saw before. All right. So again, we start with the explanation of the topic. And if you scroll down, you'll find codes here. Okay. So these codes are given in a very stepwise manner. All right. We start with the loading the packages, then loading the data. And again, with that, you'll also find the results of those particular code that you are uh, running. Okay. And you'll find the if there are any plots, there are diagrams attached with for those plots. And again, how to color the plots, t-SNE plot, here you can see. Now, these are the only steps discussed. Okay. But you want to practice this codes and you want to learn these codes, get an explanation of these codes. So for that, we have provided links of Google Colab notebooks. Now, why Google Colab notebooks? So one benefit, it is mostly for your own convenience that we have provided these notebooks. Now, one benefit is that you do not need to install any um, external tool to run the code. You can just open these notebooks, create a copy for yourself, and practice the codes. And another benefit is that if while practicing the codes, you encounter any issue or you are stuck somewhere, then what you can simply do is share the Google Colab notebook with us by copying the link and email us so that we can assist you with your query. Okay, now if you scroll down, you'll find explanation of the particular topic that we are covering in this notebook. Then we start with the codes. You can run these codes. And for here example, you can see the result is already displayed. And again, you scroll down, You'll find all the codes, their explanation, and in the same Google Colab notebook, you'll also find assignments. So here you can see these assignments will basically include to complete the codes, or if the code is wrong, then you'll have to correct it. Okay. So again, if you, while doing it, if you encounter any problem, you can simply click share and share the code with us, the Google Colab with us. And you cannot practice the code here. So basically you can just click on new notebook in Drive, and or create a save a copy in Drive. All right. So this is what the Google Colab notebook is. And if we go back and explore the R for Omics data analysis, again, we have two courses, R coding course 1, which again is a beginner friendly course for the people who are not very familiar with coding. So we start with introduction to R and R Studio, introduction to Omics data, what are the basic concepts of R programming, packages that are used in R programming and that are specific to Omics data analysis, how to import and export the data, how to manipulate the data and explore it, introduction to charts and graphs in R, and introduction to statistics in R. Okay. And if we go back again and explore the advanced course of R, it again covers the machine learning part of the R programming. We start with data processing, advanced data visualization, dimensionality reduction, PSA and visualization, data visualization using t-SNE, statistical analysis, differential gene expression, clustering, supervised analysis, advanced supervised machine learning analysis. And if I open this lesson for you, and again, you'll find the code for this particular topic, which is differential gene expression. And at places, you'll also find interactive plots. Okay. So, and again, next, and you'll find the Google Colab notebook that is associated with the lesson. All right. Now you have learned everything. You have built a foundation of genomics, transcriptomics, or any other omics, but you want to learn the application of it. Okay. So for that, we uh, have a have created example projects. And for example, you are interested in knowing about cancer and how any omics field like transcriptomics or genomics uh, is used in that particular analysis of the data set. For that, you can see here breast cancer and oncogene variant calling. So this particular project will discuss genomic aspect of the bioinformatics. You can see we start with discussing the problem in the first few lessons, and then we proceed with the analysis part. I'll show another lesson, another course for you. Okay, this is. Okay, so next is TCG liver cancer precision oncology. Now this one discusses transcriptomics. Again, you start with learning about the research problem, and then we start with the transcriptomic analysis of that problem. And in this particular uh, project, we are also discussing multi-omics data integration. Okay. And what if you are interested in, now this was about cancer. And if you're interested in neurodegenerative disorders like Alzheimer's, then we have a project on that as well. And in this, we will discuss about differentially expressed genes in Alzheimer's, which again is a course that example project that will employ RNA-seq data analysis techniques. And similarly, if you are interested in infectious diseases, then these are the courses that you can go, example projects that you can go through, like tuberculosis infection and treatment, host response to malaria infection, COVID-19, TCGA liver cancer, Ebola virus, and deadly mutations. Okay, so this all of that I have discussed with the uh, in the course library is just a overview of what you will be getting into. Okay, the otherwise there are lot many courses that you can explore. I believe all of you have signed up, so after this webinar is done, please feel free to explore the portal. And now sometimes you don't want to learn everything, okay, you know, want to have a targeted approach to learning, and for that we have created specialization tracks. Now we have 7 specialization tracks in total. We start with Introduction to Omics Bioinformatics, which is a basic beginner uh, level course track. I'll close these tabs quickly. All right, now I'll show you. So course tracks are basically a group of curated courses that might interest you. So in this particular track, we are introducing bioinformatics, and we have included courses like Introduction to Bioinformatics, uh, Introduction to Python for Omics data analysis, and R for Omics data analysis, and how to design a bioinformatics research project. Okay, now next course track, which is a popular course track, is multi-omics course track. Now in this uh, particular course track, we have brought 3 omics in this one track: genomics, transcriptomics, metagenomics, included Introduction to Linux for omics data analysis. Okay, so these 4 courses are included in this particular course track, along with the introductory course on bioinformatics. Now you have learned everything from this particular course track. Okay, but you want to learn about the application of it. So for that, we have a course track on NGS for infectious diseases. Okay, so this particular course track includes all the example projects related to genomics, metagenomics, and transcriptomics. We have SARS-CoV-2, origin and pathogenesis of SARS-CoV-2, Ebola virus, deadly mutations, and enterovirus, uh, surveillance of SARS-CoV-2 variants of concern, tuberculosis infection and treatment, transcriptomic profiling of long COVID. So for this particular course track, we recommend that you go through the multi-omics course track first. All right. Now we have created a course track for both R, uh, R and Python for omics data analysis, and these include, this is the Python one, basic Python course, beginner one, and advanced Python course, which includes machine learning aspects and introduction to Linux for omics data analysis. And similarly, R for omics data analysis, which has a basic R coding course, then advanced R coding course, and introduction to Linux for omics data analysis. Now, the last 2 course tracks are, one is RNA-seq data analysis for biological data science. Now, in is covering RNA-seq data analysis in detail. You start with transcriptomics course, then you proceed to learn all the example projects that are related to RNA-seq data analysis. The example projects are PDX, cancer macrophages, potato drought resistance, and Alzheimer's project. And lastly, we have is a course track on single cell transcriptomic analysis. So this one includes 3 courses. First one being single cell transcriptomics, which will introduce to all the aspects of scRNA-seq data analysis. from basic to advanced level topics. And these two are the example projects, which are the application of what you will learn in this particular course. So these example projects are tumor heterogeneity of breast cancer and single cell RNA-seq for lung cancer. Okay. Now, if you want to enroll in this particular course track, what you have to do is simply go to the page, open up a course track, and select any of these three access periods. First one being 1 month, then 2 months, and 3 months. So you can choose based on your preference. You can choose any of these three access periods. All right. So if there are any questions that you might have regarding this, we can address them in the end. So Shrigori, you can take over.
Oh, thank you so much, Jivisha. I'll share my screen now. Okay, so before I move on to the next part, just to summarize what we discussed so far. So on the platform, we showed you the technology aspect, related coursework on genomics, transcriptomics, metagenomics, R coding, Python, and so on, right? So after you're learning, how can you apply all of this using real world data set is what is covered through the example projects. And these example projects can be from any discipline like neuroscience, infectious disease, metabolic disorders, so on, right? And all of these coursework was what was discussed so far. And now if you're interested to go through a curated collection of coursework and example projects on a specific topic, in that case, you can go through these specialization tracks. Okay, so this is just to summarize. So all of this are the training resources that will help you build a solid foundation in your chosen field of interest. Now, most often you must have noticed after your training, you get a certificate, but most often when you go for job interviews or you're applying for your PhD, most often they require you to have some project or research experience where you're able to translate all of these learning skill sets that you have into a project format, right? So that is why there is a pipeline that we have built right from training through either coursework or tracks into a research project. So if you're someone with a passion for research or want to work on a project, we also have a separate dedicated research program that I would like to quickly show you through so that if you're someone who's interested to explore, then you can also learn about it, right? So what is this research program all about? So it is an unparalleled opportunity for whether you're a student or a researcher or even a citizen science to work independently on a bioinformatics research project. So with mentor support, you will be able to delve into real world bioinformatics research using omics data, right? So now how does this exactly follow along, right? So here I have put together a roadmap. So as you can see, it begins with the students undergoing a comprehensive training in your chosen field of omics. Like mentioned, it could be genomics, transcriptomics, metagenomics, metabolomics, or even cheminformatics. And the objective here is to help you build a strong foundation in key skills such as data processing, analysis, and visualization. So all of that training can be achieved through training aspect. Okay. Now, in the next stage, students will be able to work on identifying an impactful research topic and through regular mentor meetings and in-depth literature review. And finally, in the third step, students will be able to work on data collection, analysis, and interpretation. And this is also the stage where you will start looking into data set to identify trends, patterns, and correlations that will be critical later on for the research at hand. And finally, the program will culminate in students working on completing the report detailing all the analysis and interpretation they have carried out throughout the program, preparing a presentation, and submitting the work so that the mentor team can look into it and further refine your work. Right? So now a question might be, so what does a student completing the research program achieve? So over the years, we have been able to work with several students from diverse backgrounds to help them achieve their academic and professional goals. And all these students have leveraged the resources in very innovative ways to achieve their goals. And here on my slide, I'd like to show you quick examples of how different students have achieved this. So here you can see on the screen a cohort of students from Systems Medicine graduate program who are from biology background. They worked on developing machine learning projects after completing their training in Python to address various biomedical research challenges like infectious diseases, neuroscience, metabolic disorders, and so on. So I'll be showing you all these student projects on the platform in just a while as well. So this is just one example. And some of the students, if you are someone who's interested to participate in international research symposium and conferences, there have also been students who joined the research project to get this done. And sometimes students also join the program to get a publication that gives them this extra competitive edge over other students while applying for higher studies or in the job market. And not only undergraduates, masters, there are also students even from high school background who are working on research project. So here we have an example of a high school student from Mexico who worked on Alzheimer's disease project, and he later on went on to present at the Region International Science and Engineering Fair, for which he won the Potamkin Prize. And currently he is enrolled in a chemical biology undergraduate degree at Harvard University. So as you can see, the final goals and achievements are very diverse. And like I mentioned, some of the students, they join to get some research experience done that they can showcase when they're applying for higher studies, for their PhD applications, and so on. And one of the unique highlights is that faculties and researchers have also collaborated to carry out computational research work. Now, if you're someone with a knack for business and science, in that case, we also offer an entrepreneurship and business innovation track. So here's a short example of a team of industrial researchers from Nigeria, where they completed metagenomic training, and they worked on developing a microbiome test kit to improve the fertility of women in Nigeria. So these were just few examples that I wanted to show you. Now let me stop sharing my screen and take you back to the platform where I can show you where you can explore these projects. Okay, so here if you go to the OmicsLogic platform and head over to this projects tab here, it will display the various collection of student projects that have been published on our platform throughout the years, and you can have a look at it. So one of the recent project was from a master's student in a bachelor student in US. She wanted to apply for master's in research, so she worked on developing a machine learning project. And there are also several other examples that you can explore. You can use any of these tags to explore the topic of your interest. If you want to explore genomics projects, or if you want to explore based on the specialization like neuroscience or cancer research, you can check on these appropriate tags and it will display all of these projects. So there are around more than 50 plus projects that are listed. So if you're someone who's interested, you can definitely go through it. Okay. So I'll just quickly share this link in the chat box as well, so that if you're interested, you can go through it. All right. So now that is all that I wanted to cover in my part. Now I'd like to pass on the stage to Shubhana, who will guide you through the process of how to enroll and go about with the program. So over to you, Shubhana.
Thank you, Shrigori. Am I audible to you?
Yes.
Shrigori, am I audible? Yeah.
Yes.
So hello everyone. So the next question that might be in your mind is how to enroll, how to apply for these courses. So what you can do is you can simply go through our OmicsLogic website, click on the products tab. So first option is if you want to enroll for course library section, here you can explore the courses. As Javisha has already explained, there are 30 plus courses, 12 plus example projects on different domains like transcriptomics, genomics. And if you are looking to enroll for all these courses, we have this education subscription option, which is starting from $45 a month. It's a subscription-based model. If you want to enroll for 3 months, then it's for $105. and unlimited 1 year for $350. But we are offering a special discount for the webinar attendees, which will be 20% discount. So based on that discount, the pricing will reduce, and 1 month subscription will be available for $35. 3 months subscription will be available for $85, and yearly subscription will be available for $280. Now, if you are someone who is living across Indian region, so I can show you the Indian pricing as well. So we have unlimited coursing prices starting from 3,000 INR and yearly pricing up to 20,000. So for Indian participants as well, these pricing will reduce to 2,400 rupees, and for 3 months it will be 6,000, and for yearly subscription it will be around 15,999. So this will be the pricing for our yearly subscription or the education model. Now, if you're someone who is looking to enroll for specialization tracks, so our specialization tracks are, customizable. There are option to purchase for 1 month. If you're a slow learner, then there is an option to purchase for 3 months, 2 months as well. So price, since I'm already enrolled in one of the courses, so pricing will appear like this. So we have this basic track that is on Introduction to Bioinformatics. If you're new to bioinformatics, then this is a great track to start with. So it will be starting from 590 rupees, and I'll be showing you the dollar price as well. So it will be starting from just $10 a month. So it's a great option you can, start with. Now, if you want to, enroll for multiple specialization tracks, what you can do is, for example, you are new to bioinformatics, or you want to start with, multi-omics and then wants to learn the application part as well. So you can reach out to our team. After this webinar, we will be emailing you details of these tracks along with pricing options, pricing details, and someone who is looking to enroll for multiple tracks, we can offer them, discounts based on number of tracks they are choosing. Since it's a customizable plan, I won't be able to explain pricing for all. So this multi-omics track is also starting from $20 a month. And for Indian customers, it will be around 1,180 rupees. So what you can do is, now the next step is how to purchase these tracks. Simply, what you have to do is you have to click on buy now option. There will be this pop-up appeared on your screen. Here you have to enter your email details, your phone number details, your education background as you have already logged in, and then click on this pop-up and you can select a payment option depending upon your location. For example, if you are living in India, then you can choose for Razorpay. If you are living abroad, you can go for PayPal. And if both the payments is not working for you, we have bank transfer, Stripe, WeChat, and other payments option also available. So what I'll be doing is I'll be dropping the email address in the chat box and the products link so that you can review the products, and then you can reach out to a team or simply reply to the email after the webinar. And we will be sharing you the customizable pricing option as well. So I have pasted the products information in the chat box. I'll be dropping the email ID as well on which you can reach out. So any questions so far you want to discuss with our team, ask our mentors so that we can answer all those queries if you are having. Now I'll be stop sharing my screen and the stage will be open to you all. So if you have any questions in mind, you can discuss with our team and we will be happy to help all the queries you have in mind. Any questions? So Jivisha has dropped the contact information as well. So you can reach out to following numbers mentioned on the screen in your chat box. So there are 3 numbers, one email ID mentioned. You can reach out to anyone and our team will be happy to help.
Please feel free to ask any questions you might have from the session.
Yes, so Kavya has one question on research-based courses. So I'll be again sharing my screen. I'll be sharing you the research-based courses that we have. So you can simply go to specialization tracks. And then here we have is infectious disease course, which is a research-based course. There are multiple research projects available for you to go through. So all the course, you can see the course library towards the bottom. It has SARS-CoV-2 Ebola virus course. So the pricing is starting from 1180 rupees for 1 month. If you're a slow learner, you can enroll for 2 months because every individual's learning pace is different. That's why we have kept these three duration: 1 month, 2 months, and 3 months. Access will remain same. Coursework will remain same. For example, you will be having access to all the courses for the duration of 1 month, 2 months, and 3 months. So these are the pricing. I'll be pasting its link in the chat box itself so you can access it. Similarly, I'll be showing the dollar price as well. So this infectious disease track is starting from $15 a month. Any other queries from any participant? We will be happy to answer. I hope, Kavya, your question is answered. If you have any other query, please feel free either to unmute yourself. Niche topics with mentor guide. So Kavya is asking for the course path. So Kavya, for that, what we have is based on individual's profile and different projects they will be choosing. We have a customizable plan for them. What we can do is we can drop you an email with a customized plan that you have based on your profile, based on the project you will be choosing. We can share a plan with you so that you can decide accordingly and start your research journey as well. Okay, thank you, Kavya. Anyone who want to unmute themselves or paste their queries in chat box, stage is open for them. Thank you, Swan. So we look forward to everyone who is attending this webinar to join these courses and start their bioinformatics journey. Jeez. After completion of course, are these projects that we have to work on or get recommended? Or is it so we will be sharing the recordings as well after the webinar. So Vedanshi has a question. Okay, after completion of courses, are there projects that we have to work on or get recommended? So these projects that we have these are for learning purpose. What you can do is, for example, the multi-omics track, that is the learning track, and the application-based track is this one, the infectious disease one. Whatever you have learned in the multi-omics track, you can apply your knowledge on these tracks and practice how to, build structure a research project that will be the outcome of this particular track. Thank you, Sabha. So once the recording is processed, you will receive the recording via email. So I'll stop sharing my screen. And in the meantime, if anyone has any queries, please feel free to paste their queries in chat box.
Yes, Amit, you can definitely use these projects. resume. You mean example projects or research one? Because for each of the course that you complete on the portal, you'll get a certificate for that. For any course, if you're completing genomics, you'll get a certificate for that. If that is transcriptomics, a certificate for that. And if any example projects, like I showed you, you'll get a certificate for that as well. And if you are going for a research, then with us, then you'll have a certificate for that as well after the completion of your research. And for the course tracks, you'll have certificates for each of the course that you complete, and then combined track certification as well. I hope that clears up the query, your question. All right. Any more questions?
Uh,
Okay, yeah, Shubhangna, you can answer.
So, Sumit, what you can do is we have dropped the email address as well as contact information in the chat box. I'll drop it again so that you can contact our team through email or you can contact through phone number, WhatsApp, whatever is comfortable with you.
I have shared, Shubhangna.
Okay, thank you. So with Anshi, for placements, what we have is we do provide placement assistance. And if there are any opening in future, what we do is we have a standard process of hiring. So we go through that process, and if one is selected, we definitely help them in getting placed. So it depends on individual's effort as well. So what you can do is you can upskill yourself by enrolling to these courses.
If you have any doubts, please feel free to unmute yourself or ask them in the chat box. If not, then we can end the session. And we hope that this session was helpful for you in some way. Okay, I hope there are no further questions. So you can definitely reach out to us. We have added the numbers from our team and also the email ID. You can reach out to us however you want. There any like, ma'am, yearly monthly subscription, is there any restriction in accessing courses?
No, Kavya. So all the courses will be available under yearly or monthly subscription. It's just the duration difference. Someone who is a slow learner might enroll for a longer duration. And if you are, you think you can complete all the 30 plus courses in a duration of 1 month, since they are extensive courses. So that's why we have different durations. All the courses will be accessible under education subscription for a month. The only difference is of duration. I hope that answers your query. Okay, thank you, Kavya.
Okay, I hope there are no queries anymore. If you still have any queries, so you can reach out to us at support@omicslogic.com, or any query related to enrolling, then you can reach out to us at communications@omicslogic.com. So we'll end this session, and I hope that you all become a part of a community as well. So thank you for joining today, and have a nice day.